Today I'm gonna talking about integrated resistance measurement. Actually, this is a kind of case study because I don't know exactly what kind of insecticide are used in your country and what kind of pests are major pests in your country. So it's totally depending on environment and usage of insecticide. So please understand, this is a kind of example. So if you find some kind of insecticide resistance in your country, then please apply this kind of concept. So this is the case study. So diamide resistance status and molecular-based management strategy of Spodoptera exigua in Korea. Based on IRM mode of action classification, there is in total 5 insecticides grouped on the diamide. So diamide is a target site is iodine receptor. So this is the mode of action is that iodine receptor modulators. So it group 28. And in Korea, chlorantilanipro and flubendiamide first legislated in Korean government. After around a decade, chlorantilanipro is one of the most widely using insecticide in worldwide for the Lepidoptera pest control. So, as I mentioned, in total, 5 types of insecticide diamides. So this is an interesting review for the diamide resistance, 10 years of lesson from Lepidoptera pest. So, as this paper mentioned, widely using the diamide, particularly in chlorantilanipro, some of the reason, some kind of insecticide resistance already reported. So in this reason, particularly on Brazil, in this case, Spodoptera prosperida insecticide resistance is extremely high. So diamide resistance confirmed in Brazil and Costa Rica for the Spodoptera prosperida. And in here, China and Japan and Korea, the high level of diamide insecticide resistance was reported for the Spodoptera exigua. So until now, main Spodoptera pest, Spodoptera prosperida and Spodoptera exigua, was reported for the diamide insecticide resistance. And this is the target site of diamide insecticide, that is the iodine receptor. It's a little bit complicated forms for the iodine receptor. Anyway, the main insecticide resistance mutations existed in here. That means it's a transmembrane segment here. So, in total, 3 point mutations were reported. So, this is the first aim on this research. Diamide insecticide susceptibility of Spodoptera exigua on field population. So I was checked the Spodoptera exigua insecticide resistance on 2019 against 21 insecticide. So as you can see in this figure, most of diamides showed when we treated on the recommended concentration, their mortality less than 50%. So except tetranilipro, but the tetranilipro is also less than 60%. So that means, except tetranilipro, when we treated this kind of diamide insecticide in a field, only less than 50% larva can be controlled. So after treatment, alive larva can grow up and then population will be dramatically increased. So that is the problem. But what about other insecticide? For the fluoxametamide and bruflanilide and indoxacarb, these kind of insecticide, their efficiency is really, really good. So as a result, if there is some Spodoptera exigua in your field, then I recommend this kind of insecticide. So let's think about this like this. If there is some kind of a pest in your field, then if we treat it just one time of insecticide, but if we using this kind of insecticide, the control efficiency is really low. But if we using this kind of insecticide, control efficiency is really good. So just when we choosing what kind of insecticide, the control efficiency is totally different. Then we have to consider again and again what kind of insecticide will be used. That is really important. So depending on the insecticide resistance, we can choose. This is the susceptibility of the susceptible and diamide resistance strain and the 6 local populations of Spodoptera exigua against 2 diamide insecticide. At that time, I was used chlorantidinipro and flubendiamide. So we said this is a cross resistance. If in this case, these chlorantidinipro and flubendiamide, their mode of action is the same. So that means if there is some kind of genetic change when it against to the chlorantidinipro, then that mechanism is the same. So flubendiamide can also show the high level of insecticide resistance like this. On the diamide resistance strain, it showed over 28,000 folds on this kind of high level of insecticide resistance based on SC50 value. SC50 means lethal concentration of 50. And anyway, when we using the same strain treated another insecticide, flubendiamide, it also showed 135,000 higher than the susceptible. So extremely high level of insecticide resistance showed. So that is the cross resistance. And also not only this resistance strain, but also other local populations showed a high level of insecticide resistance, particularly on Yeoju or other population also showed high level of insecticide resistance. This is the result of susceptibility comparisons of field populations of Spodoptera exigua larva to chlorantidinipro. At the time, we treated just one concentration, that is the recommended concentration. So on commercial products of insecticide, on their label, the recommended concentration is noted. So that means if farmers using this commercialized insecticide, then if farmers use this concentration, then you can expect at least more than 95% mortality. But yeah, this is the result. When we treated on recommended concentration, but on 2015, at that time, we treated the same concentration. The mortality, most of field population showed 100%. Their control efficiency was really good. But after 3 or 4 years, 2017, most of field population showed high level of insecticide resistance. It cannot control in this recommended concentration. But some of reason can show 50% mortality. So it depending on seasonal and the yearly based. There is some kind of variations. And we try to find out what is the main factor and what kind of genes involved in this insecticide resistance. So we make a F1 hybrid. This means resistance male and susceptible female cross F1 hybrid. And here is SR means susceptible male and resistance female to make a F1 hybrid. When we compare two groups, there is no statistically different. So that means some kind of insecticide related genes located in a autosome, not a sex linked. And we try to find the degree of dominance, that is minus 0.7 to minus 0.5. So on Chinese group is also identify the degree of dominance. Only the I to M mutation existed in a resistance strain. At that time, they showed the degree of dominance minus 0.9. So that means in Korean population, probably it linked to just not only one gene. So many of genes can be involved in insecticide resistance than Chinese resistance strain. And also F2 screening was conducted. Strategy is that to make a F1 hybrid crossing the susceptible and resistant strain, after mixed together to make a F2 hybrid. So on a F2 hybrid contains some of susceptible earlier and the resistance earlier, and then we check one by one. So this is the summary of these results. Bioassay based insecticide resistance monitoring. So based on the bioassay, we can select 6 insecticides with excellent insecticidal effect. So fluoxametamide, bruflanilide, this kind of insecticide, this is a metadiamide group. It's really effective. So in this case, I can suggest for the farmers, please use this kind of insecticide to control the Spodoptera exigua. Please do not use this kind of insecticide because it showed high level of insecticide resistance. So even though if you spread this kind of insecticide, their control effectiveness is really low. And chlorantraniliprole based diamide resistance states of Spodoptera exigua, so resistance levels were dramatically increased from 2017 to 2015 to 2017. So that means during this time, probably there is some kind of genetically changed. So resistance level was varied by local and year in various local populations, and cross resistance was confirmed between the chlorantraniliprole and flubendiamides. And the identification of diamide resistance mechanism using linkage analysis. So to make a F1 hybrid or to make a F2 hybrid, that is linkage analysis. So through F1 hybrid analysis, it was confirmed that resistance-related gene existed in autosomal loci, not a sex-linked, and inheritance by incompletely recessive form, or maintains considerable resistance even in the hetero form. So let's focusing on what kind of changes in a gene level. So this is the aim 2, identification of diamide resistance mechanism in Spodoptera exigua. We were checked, already reported some of research results. So on the Chinese group reported molecular identification of 4 novel cytochrome P450 genes related to the development of resistance of Spodoptera exigua to chlorantraniliprole. So based on this research paper, we can get some kind of idea about some of CYP9 genes can be involved in insecticide resistance. And also, using the CRISPR-Cas9, G2E mutation could be involved in higher level of insecticide resistance. So, based on this concept, we will try to find what kind of genes are linked or involved in insecticide resistance. So I was used three groups. There is one of the susceptible strain and diamide resistant strain and F1 hybrid in between the two strains, and their expression pattern is grouped like this, and then their expression pattern was checked and tried to find some kind of candidate. Finally, we find out some kind of insecticide resistance related genes like that, and this one is also. Next, we try to find out some kind of insecticide resistance specific mutations in the genome level. So there is no reference genome, so we try to set the new reference genome for the Korean resistance strain. So this is the result. Isolate BAW-KOR-DIRS1. So that means beta-amine worm, Spodoptera exigua, Korea, diamide resistance, and susceptible F1 hybrid. So we set the whole genome dataset, and then we try to find these kind of mutations existed or not. As a result, only one point mutation, M2I point mutation, was identified. And this mutation existed in a cDNA level and genomic DNA level, and we checked. And also, there is some kind of indel insertion or deletion existed in a resistance strain. So we checked this kind of resistance specific indel in the cDNA level and genomic DNA level. Yeah, that perfectly matched. And here is the resistance specific insertion. So based on this sequence, we set the species diagnostic primers. So here is the mutation. So this resistance specific insertion was linked to this I2M mutation. So we try to make some kind of diagnostic method. So this is the result. So using LAMP, loop mediated isomer amplification. So when we using this kind of method, we can easily identify the insecticide resistance earlier. So this result is how to increase their efficiency. Then this is the identification of the detection limit of genomic DNA in the LAMP assay. And also we applied if we using some kind of tissue, from some kind of tissue can be identified this resistance specific earlier. Yes, it was. So when we using the larval tissue or adult tissue is also, we can easily distinguish this is the resistance and this is the susceptible. This is resistance, this is susceptible, like this. So that means if we using this kind of diagnostic technique, then before treated insecticide, after this kind of simple technique, then we can check the insecticide resistance existence or not, like this. When we apply the local populations, so untested local population showed has all resistance earlier, and also it can modify like this multiplex PCR. The multiplex PCR result is also almost the same result on a lamp. So this is a kind of good example how to diagnose the insecticide resistance and how to select the effective insecticide. So this is the results. To find the insecticide resistance mechanism and to develop this kind of molecular-based insecticide resistance diagnostic tools, that is not so easy. Yes, I knew that. But if you can apply these developed molecular tools, then that could be possible, I think. So, well, depending on your country, this kind of molecular-based insecticide resistance monitoring tool can be applied. So here is the diagnosis process summary for the lamp. First, sample preparation. React the PCR tube containing 30 microliter nucleus-free water and a part of larva or other tissue at incubate at 95 degrees of Celsius 5 minutes. Then DNA will be released. And next step is lamp. Add sample, so supernatant of this 3 microliter of supernatant, transfer the tube, another tube, and mix lamp reagent, primers, and react at 61 degrees of Celsius for 90 minutes. Then we can check the result. Diagnosis confirmation, insecticide resistance, it turned to yellow, and susceptible will be pink. So it's really simple, and it takes less than 2 hours. So if we check the insecticide resistance, then we can choose more effective insecticide. So that is kind of case study. So if you are interested in these tools, then please apply your country. Thank you very much.